Now we're going to be covering the next set of slides from slide 31 to 40. So what is a clad? A clad is a clade. A clade is a group of organisms consisting of a common ancestor and all its descendants, representing a complete branch on the tree of life. For example, the clade Escherichia includes E. coli, E. alberti, E. fergusoni, all species descended from the last common ancestor of the genus. So clade is basically a group of organisms consisting of a common ancestor and all its descendants. Clades can exist at any taxonomic rank, genus, family, order, etc. And in metagenomics, identifying clades helps place new organisms on the tree of life, even if they are only known from sequence data. Now the taxonomic hierarchy. So the classical hierarchy, domain, kingdom, phylum, class, order, family, genus, species, provides the framework for naming organisms. Metagenomics often discovers DNA fragments that don't fit neatly into existing groups, leading to the creation of a new higher taxa, that is new families, phyla, etc. This reshapes the tree of life dynamically. Metagenomics isn't just filling in species but rewriting major branches. Now the metagenomics discovery pipeline. So discovery process typically looks like this. Firstly, we do sample collection and sequencing. Then we assemble all of these into contigs, right? Then annotation of genes and functions, and finally database deposition for community use. So this pipeline ensures that newly found organisms are integrated into a broader reference system, allowing future metagenomic datasets to classify similar organisms more easily. Case study: Global Ocean Survey. The Global Ocean Survey was a landmark metagenomic expedition led by Craig Venter, sampling seawater across oceans. Results: tens of millions of new genes, thousands of microbial species, and hundreds of novel taxa never described before, revealed that ocean microbes are the engines of global biogeochemistry and that most were invisible to culture-based microbiology. This case proved the power of metagenomics for biodiversity discovery at planetary scale. Now, moving on, sample to database flow. After sequencing, environmental DNA is annotated, taxonomically classified, and deposited into curated databases like NCBI, RefSec, ImageM, MGRAST. This process builds the reference infrastructure that allows later metagenomic studies to do faster, more accurate classification. Without these community databases, each study would be isolated, but with them, global knowledge accumulates, moving beyond who is there. Once basic taxonomic presence is established, the next question arises: how abundant are different organisms? How does this vary across time, space, and environments? And how unique is one community compared to another? These questions lead into concepts of diversity and uniqueness, which are central to ecological and biomedical interpretations of metagenomes. Now, the community diversity. Diversity refers to the richness, how many species are present, and the evenness, how evenly distributed their abundances are over a given area in a community. Metrics: Shannon diversity, Simpson's index, Chow-1 estimator. Example: gut microbiome diversity is higher in non-Western rural populations compared to urban Western populations, correlating with diet and health outcomes. Right? Diversity is often used as a health indicator. Loss of diversity is linked to disease. Now, community uniqueness. Uniqueness asks how different one community is from another. Beta diversity. Metrics: Bray, Curtis, dissimilarity, unifrac, phylogenetic based. For example, vaginal microbiomes dominated by lactobacillus are distinct from gut microbiomes dominated by firmicutes and bacteroidetes, whatever that means, whatever that is. In ecology, uniqueness allows comparison of biomes. In medicine, it distinguishes healthy versus diseased microbiomes. Diseased. Up to now, we've established the motivation for metagenomics: culture-independent discovery, community context, diversity, and uniqueness. And then after this, we'll be moving into the practical implementation. So first, getting the data, DNA extraction. The first step in any metagenomics workflow is extracting the DNA from the sample, that is the soil, gut, water, skin, etc. DNA extraction must break open diverse cell walls, bacteria, archaea, fungi, and viruses. Avoid bias. Gram-positive bacteria are harder to lyse than gram-negative, and yield sufficient clean DNA for sequencing. Extraction biases can skew the observed community, so protocols are critical. Now, from Y community level to what do we measure? So traditional pure culture genome function workflows systematically miss most microbes and distort the rest because one, many taxa don't grow in lab media, and secondly, genomes and phenotypes shift outside their native networks. Metagenomics fixes both by sequencing directly in the habitat, bypassing cultural and preserving community context, interactions, selection pressures, etc., which is where functions actually emerge. In short, profile the community in situ or risk a biased picture. Now, metagenomic investigation workflow, the backbone you'll reuse. A standard pipeline is sampling, DNA sampling, community DNA extraction, library prep, sequencing, base calling or quality control, then taxonomic and functional profiling, then comparative association analysis. Notice how every step can introduce bias. For example, extraction favors some cell walls, library prep alters the GC representation. So, reproducible protocols and appropriate algorithms here are as important as the sequencer itself. Four foundational definitions you will reference constantly. So, firstly, we have genome, the full DNA of one organism. Then we have metagenome, the aggregate DNA from all organisms in a sample or a community. Microbiome, the organisms plus their genomic functional content in an environment. And the pangenome, the union of all genes across all strains in a species or a clade, reminding us that strain level diversity drives function. These definitions clarify why community data is not equal to many isolated genes. It's one ecosystem scale data set. Now, who is there is the first metagenomic question. Before anything else, we must identify which clades and species are present and their relative abundances. That implies mapping anonymous reads to a taxonomy that spans domain to species. The hierarchy matters because resolution determines interpretability. For example, genus level patterns may miss strain linked traits such as virulence or resistance. Species and clade, why these labels are analytical levels. Species are a biological concept, and clade is evolutionary lineage on the tree, and are not just labels. They determine how we aggregate evidence. Signals pooled at the wrong level blur real biology. For example, mixing commensal and pathogenic E. coli. Clear definitions prevent that analytical smearing. From unknown organism to characterized species, the discovery ladder. The practical ladder is assemble a draft genome, annotate genes, place lineage on the tree, and then functional type with pathway predictions, then experimentally test the hypothesis. The handoff to reference databases is critical. It turns a one-off discovery into reusable knowledge for future datasets. Unknown has levels, genus to phylum, and each level changes strategy. Novelty can be shallow, new species in a known genus or deep new phylum. The deeper the novelty, the less helpful reference-based classification becomes, and the more you need assembly binning plus de novo annotation to infer biology. Expect uncertainty to grow with taxonomic distance from references. Case study, Global Ocean Survey proves the point at planetary scale. Now, large-scale environmental sequencing of ocean water revealed around 1,800 species with 143 previously undetected, later expanded across 23 countries to more than 1,000 more unseen species. The lesson is that culture-free metagenomics rewrites biodiversity baselines and exposes the true tree of life breadth in key biogeochemical systems. Beyond presence or absence, diversity and uniqueness. So after who, we need to know how they're represented. Alpha diversity, richness, evenness inside a sample, and how communities differ. Beta diversity, between sample uniqueness. These metrics underpin ecological and clinical inference. For example, this biosis signals biome clustering, so they're not optional. They're the core readouts of a metagenome survey. Now, yeah.